a crazy amount of cost. And so I went down this rabbit hole of normobaric hyperoxia, and I wanted to share it, what I learned, in a couple of videos. This is going to be part one of this. And then part two is actually going to be even more shocking, at least from my perspective. So without further ado, let's just keep moving forward. So the first paper I pulled was from Med Hypotheses in April of 2011. The title was called The Normobaric Oxygen Paradox, a novel way to administer oxygen as an adjuvant treatment for cancer, ? And what it says here in the abstract is the normobaric oxygen paradox is a dual mechanism by which oxygen regulates the expression of the hypoxia-inducible factor HIF-1 alpha. And the HIF-1 alpha depending gene regulation is responsible for many different genetic expressions, including EPO, which is erythropoietin, and VEGF, vascular endothelial growth factor, that are usually expressed in parallel. First, VEGF underexpression could decrease tumor angiogenesis, leading to a decrease in tumor growth or even apoptosis in cancer cells. Second, induction of EPO expression can provide cytoprotection, although this could be deleterious for cancer cells while helping non-malignant cells, or at least neuro, brain, and cardiac heart cells, to be protected from side effects of chemotherapy. Eventually, HIF induction could boost immune response by inflammatory cells, increasing their anti-tumor activity. So basically I pulled this because this was just kind of like just being thought of as, could this even be a thing? And so I'm going to advance our understanding by pulling this paper from May of 2018, and it was titled Normobaric Hyperoxia Inhibits the Progression of Lung Cancer by Inducing Apoptosis. And I highlighted some things in the abstract here. It says, following a tail vein injection with Lewis lung carcinoma cells, so they inoculated these mice with this lung cancer model, right? And then what they did was they exposed them to 24 hours of normobaric oxygen and hyperoxia, this cycle, for 2 weeks. And then in addition, this A549 lung cancer cells were incubated in normobaric hyperoxia chamber for 24-hour period. And as a result, the size and number of tumors in the lungs decreased significantly with exposure to normobaric hyperoxia in the mouse model. And it later says that the study demonstrated that the role of normobaric hyperoxia in inhibiting lung cancer, normal tissues and cells showed no significant hyperoxic damage in our experimental settings. The anti-tumor effects of normobaric hyperoxia may be due to the increased reactive oxygen species activity and apoptosis, which is related to the mitogen activated protein kinase pathway, MAPK pathway. Impact statement: normobaric hyperoxia is a feasible therapy for cancer with low complication rate. Although NBO, normobaric oxygen, may be beneficial in cancer treatment, very few studies have been conducted. Thus, the evidence is thin. This is the first study to clearly demonstrate morphological changes in lung cancer with normobaric oxygen exposure to investigate the underlying mechanisms both in vivo and in vitro. And basically what they did was they put these mice in cages, like a glass cage that was exposed to, when they wanted the mice to be exposed to oxygen, 95% oxygen. And there's other times when they would, these 24-hour cycles where they would put them under normal conditions. So let's see what actually happens to the mice that had oxygen versus the controls. So the average number of control mice tumors after injection with these cancer cells was about, let's say, 21. And if it was the mice who got oxygen at 95%, they had less than half of the amount of tumors. And then when they looked at actual tumor volumes, the tumor volume was about 50 for the control mice. And if the mouse got oxygen therapy, just oxygen therapy, this is not with chemo, this is not with radiation, this is oxygen only, then the tumor radically shrank down to about 5 to 10%. So I found this to be extremely interesting. And so it then kind of like looked at, well, what could be going on, right? And I think that the hypothesis always with hyperoxia, whether it be HBOT or ozone or other hyperoxic therapies, is that you're increasing reactive oxygen species compared to baseline, right? So you see here that at baseline, and I believe this is going to be a normal